Here is your Exam 2 study guide with additional relevant concepts inserted directly into each section. Additions are clearly marked with new and integrated where they naturally fit. Study Guide for Exam 2, Expanded. BL 223. No study guide is perfect. This study guide is intended to do exactly that, guide your studying. This is by no means a complete or exhaustive list. Please make sure you are comfortable with all items assigned and presented during our time together. And of course, please come see me in office hours with questions. Comprehensive briefing on plant physiology, defense, and environmental adaptation. Executive summary. The documentation provided offers a high-density analysis of the mechanisms governing plant growth, survival, and interaction with the environment. Central to these processes is a complex network of plant hormones including abscisic acid, ABA, brassinosteroids, oligosaccharins, auxins, and cytokinins which regulate development from seed dormancy to maturity. These biochemical signals coordinate responses to light, phototropism, gravity, gravitropism, and physical touch, thigmotropism. Equally critical is the plant's multi-layered defense strategy. This includes physical barriers, dermal tissue, a sophisticated chemical arsenal of secondary metabolites, e.g., alkaloids and tannins, and systemic responses that prime the entire organism against pathogens. Furthermore, the documents detail the essential nature of soil nutrition, highlighting symbiotic relationships with nitrogen-fixing bacteria and mycorrhizal fungi, while addressing the modern challenges of rising atmospheric CO2 and soil pollution. The potential for human benefit is also emphasized, particularly through the use of plant-derived compounds like paclitaxel Taxol for cancer treatment and the application of phytoremediation to decontaminate industrial sites. 1. Hormonal regulation of growth and development. Plant development is mediated by a suite of hormones that often act antagonistically or synergistically to respond to environmental stimuli. Primary plant hormones and signaling molecules. Hormone primary characteristics key roles. Abscisic acid, ABA plant stress hormone, synthesized in vascular parenchyma companion cells, regulates seed dormancy, inhibits growth, and triggers stomatal closure during drought. Brassinosteroids polyhydroxylated steroid hormones synthesized from campisterol, boosts seedling growth, cell elongation, and xylem differentiation, promotes dark growth. Oligosaccharids bioactive short-chain polysaccharides, 310 monosaccharide units, derived from cell walls, acts as signal-based regulators and structural modifiers. Auxins growth coordinating hormones, drives cell elongation and maintains seed dormancy, redistributes to shaded sides of plants. Cytokinins promoters of cytokinesis, cell division, promotes seed germination, counteracts ABA, mimics light-induced development in the dark. New additional hormones, often tested. Hormone key roles. Gibberellins, GA, promote stem elongation, seed germination, and fruit development. Mutants deficient in GA show dwarfism, e.g., green revolution wheat. Ethylene A gaseous hormone promotes fruit ripening, leaf abscission, dropping, and senescence aging. Works antagonistically to auxin in some contexts. Salicylic acid, SA, key signaling molecule in systemic acquired resistance, SAR, against pathogens. Jasmonic acid, JA, central to wound response and defense against herbivores, activates proteinase inhibitors. Strigalactones inhibit branching, shoot outgrowth, also signal to mycorrhizal fungi. New hormonal crosstalk, exam tip. ABA versus gibberellins. ABA promotes dormancy. Gibberellins break dormancy and promote germination. Auxin versus cytokinins. Auxin promotes root growth. Cytokinins promote shoot growth. High auxin-cytokinin ratio right arrow roots, low ratio right arrow shoots. Ethylene versus auxin. Ethylene promotes abscission, auxin delays it. When auxin levels drop, ethylene triggers leaf drop. Role in environmental tropisms and morphogenesis. Phototropism. Auxins accumulate on the shaded side to drive elongation toward light. Brassinosteroids positively regulate this via polar auxin transport. Conversely, ABA inhibits bending by suppressing asymmetric auxin distribution. Gravitropism. Auxins accumulate on the lower side of roots, stems to oppose gravity. Cytokinins act as an anti-gravitropic signal to balance this. ABA and brassinosteroids function as negative regulators of gravitropism in specific tissues, roots and shoots, respectively. Thigmomorphogenesis, touch response. ABA accumulates upon touch to aid in drought resistance via stomatal closure. Brassinosteroids change cell wall rigidity to encourage growth in new directions. Oligosaccharins facilitate the untethering of cell wall microfibrils and activate touch genes, TCH. New other tropisms. Tropism, stimulus mechanism. Phototropism, light auxin redistribution to shaded side. Gravitropism, gravity statoliths, starch granules settle to bottom of cells, auxin redistribution. Thigmotropism, touch changes in cell wall rigidity, calcium signaling. Hydrotropism, water roots grow toward moisture, ABA involved. Chemotropism, chemicals pollen tubes grow toward ovules, calcium gradient. Mutational effects. ABA hypersensitivity. Mutants like ARA1, protein farnesyltransferase mutation, and AB1 mRNA cap-binding protein mutation show reduced water loss and increased seed dormancy due to rapid stomatal closure. Brassinosteroid deficiency leads to dwarfism, thickened shoots, short internodes, and compact, curled dark green leaves. Auxin cytokinin mutations frequently result in stunting and dwarfism. TCH4 mutant affects cell wall modification genes, leads to abnormal growth under stress, but appears visually normal in stress-free conditions. New gibberellin mutants, classic example. Green revolution dwarf wheat. Mutations in gibberellin signaling or synthesis produce short, sturdy plants that don't lodge, fall over. This allowed high-yield agriculture with heavy grain heads. 2. Plant defense systems. Plants utilize physical, chemical, and biological strategies to combat abiotic threats. Fire, weather, and biotic threats. Viruses, fungi, animals. Physical and dermal defenses. First-line defense. The epidermal layer secretes wax to prevent water loss and deter attacks. Protective substances. Above-ground parts are covered in cutin, while subterranean organs contain suberin. Structural barriers. Silica inclusions, trichomes, bark, and thorns provide mechanical protection. Vulnerabilities. Parasitic nematodes use sharp mouthparts to penetrate cell walls, sometimes forming root tumors, knots. Fungi utilize stomata or form adhesion pads and haustoria to siphon nutrients without penetrating the plasma membrane. New induced physical defenses. Some plants produce callose, a polysaccharide, to plug plasmodesmata and seal off infected cells, preventing pathogen spread. Lignin deposition is rapidly increased at wound sites to create a physical barrier. Chemical defenses and secondary metabolites. Metabolic pathways are often modified to produce toxins that deter herbivores. Alkaloids include caffeine, nicotine, cocaine, and morphine. Many alkaloids are neurotoxins that target herbivore nervous systems. Tannins inactivate proteins in the attacker's gut. New high tannin concentrations make leaves taste bitter and reduce digestibility. Oils, found in the mint family, repel insects with strong odors. Defensins, small, cysteine-rich antimicrobial peptides with ancient origins in innate immunity. New classes of secondary metabolites, exam framework. Class examples function. Alkaloids, nicotine, caffeine, morphine, cocaine, neurotoxins, deter herbivores. Phenolics, tannins, lignin, flavonoids, protein binding, structural support, UV protection. Terpenoids, essential oils, rubber, taxol repellents, antimicrobial, medicinal. Glucosinolates, mustard oil, wasabi, Horseradish toxic breakdown products when tissue is damaged. Cyanogenic glycosides almonds, cassava release HCN, cyanide when metabolized. Notable plant toxins. Cyanogenic glycosides release lethal cyanide HCN only when metabolized by an attacking animal. New cassava manioc is a staple food in Africa. It must be processed to remove cyanogenic glycosides before eating. Ricin, an alkaloid from castor beans. A single ricin molecule can inactivate 1,500 ribosomes per minute by cleaving adenine from rRNA, effectively blocking protein synthesis. New ricin is one of the most toxic plant compounds known. No antidote exists. Biological and systemic responses. Mutualism. Acacia trees provide food and shelter to ants, which in turn attack herbivores like katydids. Parasitoids. Wounded leaves release volatile compounds that attract female parasitoid wasps. The wasps lay eggs in caterpillars. The hatching larvae eventually kill the host. These volatile compounds are called herbivore-induced plant volatiles, HIPVs. Wound response. Triggered by injury, plants produce cysteamine, an 18-amino acid peptide, which travels to cells to produce jasmonic acid, JA. JA activates defense genes like proteinase inhibitors that bind to herbivore digestive enzymes. Systemic acquired resistance, SAR. A broad-range, long-distance response likely induced by salicylic acid, preparing the plant for future attacks. New SAR versus ISR. Critical comparison, exam favorite. Feature systemic acquired resistance, SAR-induced systemic resistance, ISR. Trigger pathogen infection, biotrophs, beneficial soil microbes, rhizobacteria. Primary signal salicylic acid, SA jasmonic acid, JA, and ethylene. Response, broad-spectrum antimicrobial broad-spectrum, induced defense. Timing slower onset, long-lasting faster onset. Marker genes pathogenesis-related, PR. Proteins different set of defense genes. New hypersensitive response, HR. When a plant detects a pathogen, it may trigger programmed cell death at the infection site. This kills the pathogen by depriving it of living tissue and confines the infection. HR often produces visible necrotic lesions, dead spots on leaves. This is mediated by R genes, resistance genes, that recognize specific pathogen molecules. 3. Soil nutrition and nutrient acquisition. Plants depend on the highly weathered outer layer of the Earth's crust, which contains 92 naturally occurring elements, for essential nutrients. Essential nutrients. Macronutrients 9. Carbon, oxygen, hydrogen, the primary components of organic molecules. Nitrogen, potassium, calcium, magnesium, phosphorus, and sulfur. Micronutrients 7. Chlorine, iron, manganese, zinc, boron, copper, and molybdenum. New micronutrients are required in trace amounts but are still essential. Deficiency causes specific diseases, e.g., iron deficiency right arrow chlorosis yellowing. Availability. Minerals must be dissolved in the water within soil pores to be accessible. Soil particles typically have a negative charge, attracting positive ions, K+, Ka2, Mg2, etc., which the plant must acquire through active transport. New soil charge and cation exchange capacity, CEC. Important. Clay particles and humus, organic matter, are negatively charged. They attract and hold positively charged ions, cations like K+, Ka2, Mg2, NH4+. Cation exchange capacity, CEC, is the soil's ability to hold and exchange these cations. Plant roots release H+ to displace cations from soil particles, then take them up via active transport. Negatively charged ions, anions, like no nitrate, are not held by soil and leach away easily. This is why nitrogen fertilizers are often lost from agricultural fields. Special nutritional strategies. Nitrogen fixation. Plants cannot convert atmospheric N2 into ammonia, NH3. They form symbiotic relationships with rhizobium bacteria in legumes, which house themselves in root nodules. The plant provides oxygen and carbohydrates, while the bacteria provide fixed nitrogen. The enzyme nitrogenase is irreversibly damaged by oxygen. Leg hemoglobin, an oxygen-binding protein, protects it while maintaining a microaerophilic environment. Mycorrhizae, found in 90% of vascular plants, these fungi expand root surface area to enhance phosphate and micronutrient uptake. Two main types, arbuscular mycorrhizae, AM, penetrates cortical cells, and ectomycorrhizae, M, forms sheath around root tip. Carnivorous plants, found in nitrogen-poor, acidic soils, these plants, e.g., pitcher plants, Venus flytraps, sundews, trap and digest insects using specialized leaves and secreted enzymes. Carnivory evolved independently multiple times as an adaptation to low-nitrogen environments. Parasitic plants, over 3,000 species, e.g., dodder, Indian pipe, siphon nutrients directly from the vascular tissue of other plants. Some parasitic plants, like mistletoe, are photosynthetic but still tap host xylem for water and minerals. Others, like dodder, have lost chlorophyll entirely. Nutrient deficiency symptoms. Exam recognition. Nutrient deficiency symptom. Nitrogen, N, stunted growth, yellowing, chlorosis of older leaves first. Phosphorus, P, dark green or purplish leaves, poor root growth. Potassium, K, leaf margin scorch, brown edges, weak stems. Iron, Phe, yellowing between veins, interveinal chlorosis of young leaves. Magnesium, Mg, interveinal chlorosis of older leaves, Mg is mobile. Calcium, Ca, blossom end rot in tomatoes, distorted new growth, Ca is immobile. 4. Environmental stress and global change. The impact of increasing CO2. Rising CO2 levels correlate with increased photosynthesis and growth, but result in a decreased carbon-to-nitrogen ratio. This leads to reduced protein and mineral concentrations in plant tissues, increased herbivory, as animals must consume more biomass to meet nutritional needs, potential for human protein deficiencies due to lower nitrogen content in crops. CO2 effects on C3 versus C4 plants, important comparison. Plant type response to elevated CO2. C3 plants, wheat, rice, soybean, strongly increased photosynthesis and growth, reduced protein and mineral content still a concern. C4 plants, corn, sugarcane, sorghum, smaller response, already concentrate CO2, may become less competitive as C3 plants catch up. Cam plants, cacti, succulents, minimal response, already highly water efficient. Ozone and air pollution. Ground level ozone, O3, damages plant tissues by causing oxidative stress. It enters through stomata and generates reactive oxygen species, ROS, leading to visible leaf injury, stippling, bronzing, reduced photosynthesis, and crop yield losses. Phytoremediation, environmental cleanup. Plants can be utilized to remove or neutralize soil pollutants at costs 50–80% lower than mechanical removal. Phytogradation. Contaminants are taken up and broken down, e.g., poplar trees degrading TCE into CO2 and chlorine. Phytovolatilization. Contaminants are released as vapor through the stomata. Used for mercury and selenium, contaminant is transferred to air rather than eliminated. Phytoaccumulation, also called phytoextraction. Toxic heavy metals, arsenic, lead, cadmium, are concentrated in plant shoots for later harvesting and disposal. Hyperaccumulator plants, e.g., Th. laspi caerulescens for zinc cadmium, can store 1% of their dry weight in metals. Additional phytoremediation types. Type mechanism example. Rhizodegradation microbes in root zone break down contaminants, oil spills. Pause. Rhizofiltration roots absorb metals from water, not soil. Water treatment wetlands. Phytostabilization plants immobilize contaminants in soil, preventing spread mine tailings sites. Limitations of phytoremediation. Slow may take many growing seasons. Limited to shallow root zone. Depth of contamination matters. Toxic metals in harvested plants become hazardous waste. not effective for highly concentrated pollutants. 5. Human applications and medicinal value. Secondary metabolites provide significant health benefits, though nature's supply is often finite. Paclitaxel Taxol, derived from Pacific yew trees, essential for fighting breast and other cancers. Due to low yields and the finite population of yew trees, there is an urgent need to synthesize alternatives. New semi-synthesis from precursor in yew needles, renewable, and plant cell culture are now used. Quinine, extracted from cinchona trees, an effective treatment for malaria. New quinine was the first effective malaria treatment. It tastes bitter. Tonic water contains quinine. Phytoestrogens, found in soy, may lower prostate cancer rates and minimize menopausal symptoms, though their effect on infants remains a subject of inquiry. New other medicinal plant compounds. Compound plant source medical use. Aspirin salicylic acid derivative, willow bark pain relief, anti-inflammatory. Morphine opium poppy painkiller, opioid. Digoxin foxglove heart failure treatment. Artemisinin sweet wormwood malaria treatment, modern first-line therapy. Vinblastine/vincristine periwinkle plant cancer chemotherapy, Hodgkin's lymphoma, leukemia. New ethnobotany and bioprospecting. Indigenous peoples have used plant medicines for thousands of years. Bioprospecting is the search for new pharmaceuticals from plant and microbial sources. Rainforests are hotspots for bioprospecting. Loss of biodiversity means potential loss of undiscovered medicines.